Alright class, I decided to do a video recording on this particular slide so that you guys can watch it over and over again to make yourself more understand about the transcriptions and translations in Eukaryote. Right, as mentioned in the class, where the transcription process that occur in nucleus has to go through a really tight regulations before the mRNA can be coming out from the nucleus and to be translated into a protein. So what we can see from here, double-stranded DNA, where transcriptions occur, where it will produce an mRNA, and this mRNA is still a premature, where when we look at it, we still have like exons, and intron and exon and intron and over again. So this premature mRNA has to be modified before it can be a mature mRNA. So in here we have the methionine guanine cap as well as poly A tail being added. So now we have two out of the three characteristics of mRNA, which is the cap and the poly A tail. However, this premature mRNA still has some introns in it. That's why it has to go to splicing to splice out the intron so that it will now become a matured mRNA and go into the cytoplasm and to be transcribed, to be translated into protein. So like transcriptions, translations in the cytoplasm require really tight regulations, and in cytoplasm, the one that regulate the translations, I know we all know about the translations of protein where it require ribosome and so on and so forth. The same apply, but however, this mechanism is more in depth so that you can understand more and how the virus can like manipulate the cellular protein to synthesize their viral proteins. Okay, let's begin here. First of all, we know the ribosome of the eukaryotic cell is ATS, and this ATS will be split into two, which is 60S and 40S. In order to avoid the binding of the 60S back to the 40S to form ATS again, and eukaryotic initiation factor 3 and eukaryotic initiation factor 1 and 1A will bind to the 40S ribosome. Okay, so this will bind to the 40S ribosome, and when this binding is complete, it will prevent the 60S ribosome from binding back to the 40S. Next, the next step for transcriptions is we have methionine tRNA initiator, or MET-tRNA I at the back. So this methionine tRNA initiator is very specific, where this is the one that responsible as the first amino acid that bind to the start codon of the mRNA. So in order for this tRNA to bind to it, it requires another initiation factors, which is initiation factor 2 with GTP that binds to the methionine tRNA or initiator methionine tRNA together to form a complex called ternary complex. This ternary complex comprises of methionine tRNA initiator, EIF2, eukaryotic initiation factor 2, with GTP. That's why it's a ternary complex. Right, next. Now that we have ternary complex, we also have the 40S with EIF1, 1A, EIF3, and these two complex will now combine together to form 43S pre-initiations complex. Let's have a look at how it looks like. This is the EIF3, and we have 1 here, 1A here, we have 2 here with the T RNA with methionine here. So if you remember, whenever a translations to occur, you have the tRNA that carry a amino acid to go into the A site, where then it will move to the P site to synthesize the peptide, and then acid through the E site. However, for this methionine tRNA initiators, it binds directly to the P site of the ribosome rather than the A site. If you notice carefully here, you can see that one is bound to the E and the 1A is bound to the A site. That's why the tRNA methionine initiator bind directly to the P. So now we have a 43S pre-initiation complex that is ready to bind the mRNA. However, the ribosome is huge, the cytoplasm is huge, and the ribosome is everywhere. And how does the ribosome catch the mRNA and bind it to the ribosome and allow it to translate? We have other eukaryotic initiation factors, which is IEF4, which play a role in this. First of all, we have IEF4G, 4E, 4A, and 4B. The 4E is the one that responsible of binding the mRNA cap. As we can see from here, the green one bind to the cap of the mRNA. The purple, which is the G, act as a scaffolding protein. So scaffolding protein is just like a protein that provide the site for bindings of the initiation factors. So what we can see from here, the 4E binds to the cap and the 4E bind directly to the 4G, which is the one in purple. We also have 4A and 4B that bind to the 4G, where the 4A is the one that responsible for unbinding the secondary structure of the RNA, whereas 4B is the one that responsible to bind to the IEF4G as well as IEF3. Alright, remember we mentioned about the characteristic of the mRNA, which it has a cap, an exon-only mRNA, together with poly A tails. You might wonder, why do we need a poly A tail? The next slide will show you here. Now there's a protein called poly A binding protein, or in short PABP, poly A binding protein. Now this poly A binding protein binds specifically to the poly A tail of the mRNA, which in turn will now bend the whole mRNA forward, and this protein binds specifically to EIF4G, the scaffolding protein, or the one in purple here. So by bending the mRNA, it is believed that poly A binding protein enhance the translations process of the eukaryote. There is also another protein that recently been found. It's called PIPE1, or more specifically poly A binding protein interacting protein. So it's PAIP1, poly A binding protein interacting protein. Now this poly A binding protein interacting protein 1, or in short PIPE1, responsible of binding the poly A binding protein to the EIF3 to the 4A. This is a very important protein where the scientists tried to do mutations on this protein, and they found that when this protein is mutated, the poly A binding protein, or the translations is not as efficient as the wild type. So now we can see that the mRNA is now curled back and poly A binding protein is bind to the 4G, and the complex is now ready. So what's next steps? The next steps will be the scanning of the start codon. So what happened is this 43S mRNA complex will now start moving alongside with the mRNA until they found ATG or AUG in this case. Okay. Right, the next step will be the binding of the 60S back to the 40S to allow the translations to occur. However, we have all these initiation factors that still bound to the 40S to prevent 60S from binding. The next step will be using the GTPS, which is in here. We have initiations factor 5B and 5 will now play a role in going to the 43S mRNA complex and hydrolyze the GTP of the initiation factor 2 GTP. If you still remember, we have these ternary structures. So once the 5B hydrolyze the GTP to make EIF2 and GTP, the whole complex will now dissociate. So you have the IEF3 that responsible to bind to the 40S to prevent the 60S to bind back, and now the 4G, 4A, 4B will all be dissociated and allowed the 60S to now bind to the 40S, and the translations begins. So the next translations will begin as the usual translation steps that we all know, where you have the tRNA carry the amino acid, go to the A site, and then move to the P site to synthesize the peptide bond, eventually to produce polypeptides and fall into a protein. Right, that's all for the regulations of translations in eukaryote, and I hope you understand these lectures. If you still find difficulty in trying to understand the lectures, please do come to see me. I can explain to you in more details or try to make you understand at least. And that's all.